Hi, my name is Kennedy Gale. I am a junior here at Roosevelt University, and I'm majoring in biology with minors in both psychology and chemistry. Today, we're going to be addressing the question, does sex or geography structure populations of Dicrurus Adsimilis on the African continent? On this title slide on the right, we see a beautiful picture of a common drongo, also known as Dicrurus Adsimilis, and we also have Zoe Bazilla credited on this slide. That is because Zoe has done some incredible work, and this project wouldn't exist how it does today without her. She has since graduated and moved on from Roosevelt University, but we still want to give credit where credit is due because without her, this project wouldn't be where it is today. In thinking about the plumage and color of birds, we want to think about two things, natural selection and sexual selection, and their influences on the color of birds. On the left, we see an example of natural selection in bird plumage. This owl has these brown and white feathers that make it blend in really well to the bark of the tree. That is because when owls like this owl, and other birds for that matter, are able to blend into their environment, they're harder for predators to spot. If the predators can't spot these birds, they're able to live long enough to reproduce. Upon reaching this reproductive age, they're able to mate and pass their genetic material off to offspring. These offspring, inheriting the genetic material of their parents, are going to obtain the same genes for coloration as their parents. So, birds that look like this and blend into their environment are able to pass on their genes and create more birds that look like this. Looking to the right, we see an example of sexual selection. This female on the far right has some brown on her main body that helps her blend into the bark of the tree. The male on the left, however, is very bright red and does not blend in at all. That is because while birds can live long enough to reproduction age if they're camouflaged, that doesn't necessarily mean that the females are going to want to mate with them. The females in the cardinal species are very picky and only want to mate with males that have this bright red color. So while it's enough to live to reproduction age, you still have to reproduce to pass on genetic material. And because of the sexual pressures given by the female wanting to mate with red males, the genes for this red coloration are going to be passed on to the offspring, creating a bunch of male offspring with red color. Something else that's important to think about in the context of bird plumage is that we have to look at things from the perspective of the birds. On the left, we can see a blackbird as humans would see it with the naked eye. There's a little bit of variation around the throat with some blue, but for the most part, this is a blackbird as we would know it. If we look to the right, it's kind of hard to believe that this is the same bird. There's all these bright fuchsias and blues and greens. So what's going on here? Why is the bird seeing what's on the right when we can only see what's on the left? That's because humans only have 3 cones for color vision. We have a cone to see red, blue, and green. Birds have all of these and one more. Birds can see ultraviolet light, which is essential because without the cone for ultraviolet light, you can only see the blackbird on the left. But being able to see ultraviolet light means that you can see all these bright, vibrant hues present on the bird. So when we're thinking about natural and sexual selection, we also have to think about what birds are seeing because birds are selecting based off of what they can see, not necessarily what we can as humans. This brings us to the bird in question, Dicrurus adsimilis, also known as the common drongo or the fork-tailed drongo. On the left is an image of our common drongo in flight, and on the right is a map of where these birds live, their different habitats. Different subspecies and races are grouped in these ovals, so we can see there's a blue population over here, there's a yellow one, black, orange, red, so on and so forth. These all belong to the common drongo species complex, but are different populations that have some different traits. What we want to do is take a closer look at this, and that brings us to our research question of, does sex or geography have a greater impact on the plumage of the Dicrurus Adsimilis species complex? So when we look at these birds and look at the colors of them, the colors both we see and the birds see, is it easier to explain these variations in color looking at the sex, or is it easier to explain these variations with the habitat, the geography, and that would be our natural selection factor? In order to do this, we had to collect specimens. So data was collected from 255 different bird specimens that belong to 10 different species of Common Drongo. In the bottom left, we can see all of the different museums that we had borrowed from. And on the right, you can see an image of me and Dr. John Bates at the Field Museum looking at all of our wonderful birds. There are a lot more birds than are imaged here, but here's kind of an idea of how we looked at all of the birds and collected data. As I mentioned earlier, we can't see all of the colors birds can see. So how do we know what the birds are seeing without being able to see it with the human eye? We do this through spectroscopy. Spectroscopy is a tool to be able to calculate the exact wavelength of a sample that's being absorbed or reflected. So this can be used in chemistry to look at a different sample to get the concentrations of things. Or in this instance, we're using spectroscopy to be able to see colors that aren't visible to the naked eye. On the left, you can see the setup for the Ocean View spectrophotometer. And what we do is we take our bird and we take this little pen device. And if you hold the pen device up to the bird, you can get a reading. And that reading is displayed on a graph as shown on the right. And we can tell the exact wavelengths that are being emitted and absorbed by the bird. Here is an example of one of those graphs. On the horizontal x-axis, we have wavelength in nanometers. And on this vertical y-axis, we have the reflection percent. So here, based off of the wavelength that we're seeing, we can tell what's ultraviolet light, what's visible light, and how much of it is being absorbed or reflected. And this is really important because this allows us to see what the birds are seeing and be able to get that data. We also have to get the information for geography and sex, the two things that we're comparing. So on the top, we can see specimen sex and specimen number on this label on the bird. Each bird has its own label that comes with it that gives us information about where the bird is from, what sex it is, because you can't often tell after it's been preserved, and other information sometimes about what was in its stomach, different characteristics that might have changed over time. What we want to look at right now is the sex label in that top right, and the bottom where it says West Uganda Bwamba Forest. That's where the bird was found. And what we have to do from here is go to Google Earth. Entering Bwamba Forest, Uganda into Google Earth, this is what's going to show up. A quick Google search tells us that Semuliki National Park is another name for the Bwamba Forest. So this green arrow points to where Semuliki National Park is. If we zoom in, as shown in the green box on the bottom right, we can get the coordinates of this location. This is so valuable because getting the coordinates means that we can determine where all of these birds are and we can put them on a map. We can also determine what habitats are these birds living in. Are they really forested areas? Are they very arid? Are they more woodland? We're able to get all this information from the coordinates and from those specimen labels. And from there, we can analyze our data, taking into context the sex, the geography, and the colors that we get from the UV spectroscopy. And then we go back to our question, does sex or geography have a greater impact on the plumage of the Dicrurus Adsimilis species complex? Here we're looking at sexual selection. So we have females and males separated, females being the pink dots, males being the blue dots. On this vertical axis labeled Z, we have our UV colors. And on this horizontal axis labeled Y, we have non-UV colors. And here we're able to look at the different sexes of our specimens and where they lie on this color chart. If they're higher up on the UV axis, that means they have more UV color. The further they are on this horizontal y-axis tells us what their non-UV color looks like, things that we would see with the naked eye. We split these apart because the z-axis gives us a lot of information that we can't get just looking at the birds ourselves. And here, we take that same exact data, but now we're splitting it up by forest and woodland habitats. Forest being the green dots and woodland being those tan dots. Same axis of the vertical z-axis being UV color, and this horizontal y-axis being non-UV color. And looking at these graphs separately is great at all. We can get some information from it, but what we really want to do is put them together. So on the top, we can see our graphs for sexual selection separated by females and males. And on the bottom, we see our graphs separated for natural selection from woodland habitats and forest habitats. Here, we can see that the shapes of these circles or ovals differs between sexual and natural selection when we separate by the two. When we look at sexual selection, a lot of these ovals are pretty similar. They have a lot of overlap and take up a lot of the same area on these graphs. Looking at the bottom, however, for natural selection, these green ovals are taller than the tan ovals. Remember that this vertical z-axis represents ultraviolet light. So if an oval is taller or goes higher up vertically, that means it has more ultraviolet color. So from this data, we can determine that when we separate based off of habitat, there's more variance in ultraviolet color. But we don't want to just look at ultraviolet color separated from our non-UV color. We want to look at everything together based off of the brightness, so how much is being absorbed or reflected. And when we do this, we notice there's not a lot of differences. If we look at this pink box representing the different wing samples and the data that we collect on the wings of the birds, we see that while there is some clustering for the woodlands on the right graph, and on the left graph, there's some clustering for the male specimens, clustering meaning that the data points are close together. It's happening on both sides, so there's not anything different happening in one graph versus the other. Same thing when we're looking at this rump patch, when we take the color on the rump of the bird. There's a bit of clustering that's happening, but nothing true dramatic that we haven't seen in the other graph. So we can say that there's not a lot of difference happening in the brightness of these birds. Now, when we put all of this data together, we do see that there is more of a trend for sex influencing color as a whole. So putting together the brightness, UV color, and non-UV color. Sex does seem to be the primary influence, which makes sense. Birds can sometimes be very picky, and the female birds only want to mate with the prettiest of male birds. But when we're looking just at UV color, habitat has a greater influence. This is huge because, as we said earlier, humans can't see UV light. So had we not collected this data, we wouldn't know about the natural selection influences on the plumage of these birds. And all of this just goes to show that there's a very complex relationship between both natural and sexual selection. Just because in the owl that we showed earlier, we only really saw natural selection, and in the cardinals, we really only saw sexual selection. The reality is that a lot of birds exist somewhere in the middle, with both natural and sexual pressures influencing how they look. I would just like to thank everyone who's been able to help me make this project happen. Firstly, Dr. Norbert Cordero, who is a professor here at Roosevelt University. Without him introducing me to this project, I wouldn't have been able to been on this incredible journey over the past year. I would also like to thank John Bates, who is pictured earlier in the presentation, as well as Dr. Chad Eliason, Dr. Jerome Fuchs, and of course, the Field Museum of Natural History, where all of this data was collected and this project was run. I'd also like to thank you, my viewers. I really do appreciate you taking the time to watch this presentation, and I hope you learned something new. And if anything, I hope that the next time you look at a blackbird and think it looks kind of boring, that you remember that there's more than what meets the eye, at least the human eye. Thank you so much, and I hope you have an incredible rest of your day.